Hello! Today we're going to be talking about Ebola. But before we get into how predictions and uncertainties tie in, let's review the basics. Ebola virus disease is a pretty rare but also deadly disease caused by a group of viruses called Ebola virus. Symptoms can show up anywhere between 2 to 21 days after being infected and vary from your regular flu-like symptoms all the way up to internal bleeding. About 25 to 90% of people who are infected will die. However, this number varies based on what outbreak or subpopulation you look at. Ebola viruses are transmitted through direct contact with bodily fluids and can also be transmitted to humans from wild animals, such as non-human primates and bats. But wait, was that Ebola viruses plural? Indeed. There are actually 6 species of Ebola viruses that have been known to cause Ebola virus disease. However, only 4 of these have caused Ebola virus disease in humans, and of these, the Zaire Ebola virus has been responsible for most of the outbreaks, including the huge outbreak from 2014 to 2016 in Guinea, Sierra Leone, and Liberia, as well as the current outbreak in the Democratic Republic of the Congo. There is now a vaccine for Zaire Ebola virus called Revizavizabov, or Ervebo. Back to the main point. How do prediction and uncertainty fit in? Usually, when you think of Ebola, you just think about the number of people who are infected or die. But that's just counting. If we look at diagnostic tools and outbreak modeling, we can see how important predictions and uncertainty are in combating these diseases. There are 4 main categories of diagnostic tools for Ebola virus. There's cell culture, where you isolate the virus, incubate it in some cells, and use microscopy to see whether it's actually Ebola virus. Although this will definitively tell you whether or not you have Ebola, the whole process takes days and requires lots of fancy lab and biosafety equipment. Not very practical in an outbreak. Antibody tests look at your body's response to the virus, but also aren't too useful for diagnosis because it can take a while for your body to make these antibodies. Viral nucleic acid tests and protein antigen tests look for bits of virus, like DNA and RNA, or proteins. Nucleic acid tests are more accurate but take longer, while antigen tests are not as accurate but a lot faster. For example, this rapid antigen test gives results within 15 minutes. It has a sensitivity of 92% and a specificity of 85%. This means that for every 100 people who test positive, 92 actually have Ebola and 8 don't. And for every 100 people who test negative, 85 won't have Ebola. But what about those 15 people who tested negative but actually have Ebola? This is why screening tests like the rapid antigen test are followed up and confirmed with nucleic acid tests, which have sensitivities and specificities closer to 100%. Moving on to outbreak models, although it's hard to make models to predict when the next outbreak is going to happen, we're pretty good at predicting how outbreaks unfold over time. Let's start with a basic flowchart. We begin with a susceptible population, or the number of people who could get Ebola. At some point, these people become infected and then recover. How quickly this happens depends on the infection rate and the recovery rate. This is called an SIR model and can be described using a series of differential equations. Based on these equations, you can predict the number of people in each group over time. Obviously, you can add more groups and variables to these models until it becomes messy and the differential equations become even messier. In any case, even a relatively simple model is able to describe trends in data quite accurately. For example, this model of the 1995 Ebola outbreak in the Democratic Republic of the Congo used an SEIRD model that had a 99.95% fit for the number of Ebola cases and a 98.6% fit for the number of deaths. Pretty solid. Researchers have used these mathematical models during outbreaks to help health officials predict the number of people who will get infected, how quickly Ebola spreads from one city to another, and how different interventions could reduce the spread of the outbreak. Right now, the biggest limitation to making accurate models is getting accurate data. So what now? We've seen how some predictions and uncertainties factor into diagnostic tools and outbreak models for Ebola. Obviously, there's a lot more to the story, but at the end of the day, scientists have been able to make some pretty accurate predictions with low uncertainties in order to improve how we combat and monitor diseases like Ebola.